Hello everybody, let's talk about a few interesting things. Today I'll talk about the graded potential and action potential, the differences between these two types of potential that take place in the living membrane. Then I'll talk about the functional areas in the cerebral cortex. And then I'll talk about the sensory receptors and temporal and spatial coding. So first, let's talk about the graded potential. Graded potential takes place in non-excitable cells mostly, as well as the excitable cell membranes. So this is a neuron, and we know that the resting membrane potential in a neuron is minus 70 millivolt to minus 90 millivolt. Most of the places you will see that the average resting membrane potential in a neuron is minus 70 millivolt. So let's think that this neuron's resting membrane potential is minus 70 millivolt. What's the resting membrane potential in normal resting status? If you measure the voltage inside and outside of the cell membrane, inside the cell membrane it is negative, and in the outside of the cell membrane it is positive. And how much negative? That is the resting membrane potential. So in the inner side of the cell membrane of a neuron is minus 70 millivolt negative than the outside. That is the resting membrane potential. So this is a typical neuron, and this is the cell body nucleus, and these are the dendrites. We know that dendrites have branches and multiple dendrites in a typical neuron, and this is the axon, and axon is covered by myelin sheath formed by the Schwann cells, so myelinated axon. Okay, and this part is the axon terminal, and this area where the cell body, this is the cell body, is connected to the axon. This is called the axon hillock or trigger zone. So axon hillock, which is also known as the trigger zone. Okay, so now if I stimulate this neuron with an excitatory stimulus, so if I present an excitatory stimulus to the dendrite here, what will happen? The sodium channels will get opened. Okay, so sodium influx will occur. So sodium ion, which is positively charged, that means cation, sodium will get in. Okay, because the concentration of sodium is higher inside, higher outside than the inside. So by concentration gradient, the sodium, since the concentration is higher outside, sodium will get into the dendrite. And when the sodium will get in, what will happen? The positivity will increase. That means if this is the resting membrane potential, which is minus 70 millivolt, Okay, and this is the threshold. Usually the threshold of a neuron is around minus 50 millivolt. So let's say minus 50 millivolt. Okay, that means the resting membrane potential is more negative than the threshold. So this is the threshold. Okay, and when the sodium will get in, sodium is positive, so the resting membrane potential will start to move towards positive, right? And for example, after some sodium influx took place, or sodium entered into the neuron, here from minus 70 you got minus 65. Here you got minus 60 if the stimulus is stronger. If the stimulus is more stronger, it will go to minus 55. Okay, but to stimulate the trigger zone, which is the axon hillock, here you have the ion channels. Those are highly sensitive. Okay, so if the membrane potential goes up to minus 50, because that's the threshold, right, minus 50, then these channels will get opened. That means the trigger zone will be activated. Okay, so by minus 50, which is the threshold. Okay, makes sense? So if the stimulus is not strong enough to move up to minus 50 here, then it will be like, if it is, for example, 60, it will go up to 60, minus 60, and will come down. If you stimulate a little bit stronger, by little bit stronger stimulation, then it may go down to minus 55. But if the membrane potential does not reach to minus 50 millivolt, the action potential will not be generated. So these are considered as graded potential. No action potential will be generated. The graded potential is sub-threshold depolarization or excitatory sub-threshold potential. Makes sense? So these are weak depolarizations and will not spread too far, will be very localized. So graded potential is sub-threshold and will be localized. Potential will not produce action potential and will die quickly. Now, graded potential, as I showed you here, if you increase the strength of the stimulus, then it will be stronger. So weak stimulation, a little bit stronger, then further stronger. As you increase the strength or intensity of the stimulus, the potential will increase. Make sense? Like here you see, by weak, minus 65. If you increase a little bit, minus 60, then increase further, minus 55. Okay, but will not reach to minus 50. So the membrane potential will not reach to the hillock or trigger zone. Okay, It is like, in a pond, if you throw a stone, where the stone hits the water near that area, the waves are bigger, right? And as the waves move away from that point, the waves gradually get weaker and diminishes. So that's like the graded potential. Okay. Now, if the stimulus can move the membrane potential to the threshold, like here, you give the stimulus here from the resting membrane potential, the membrane potential can reach to the threshold -50, then the ion channels at the trigger zone or axon hillock will be activated and open abruptly. That is called the voltage gated sodium channels. Okay. Now in this case, what will happen? Huge number of sodium channels will get opened. Okay. And when the sodium channels will get open, a lot of sodium will get in quickly. Okay. So sodium, huge amount of sodium will get in. Okay. And when the sodium will increase inside the axon, those sodium will further open more and more sodium channels. So a lot of sodium will quickly get in, and the membrane potential will move very fast and will go all the way to the axon terminal. Okay, and in this case, the strength of the membrane potential will not get weak, will stay the same. So in this case, when the membrane potential will reach to -50 threshold, then suddenly huge sodium influx will take place. So that will move the membrane potential towards more positive. And usually in case of a neuron, it moves the membrane potential up to +30 millivolt. So that's a lot, right? So from -50 to 30, that means 80 millivolt positive, 50 plus 30, right? From 50 to 0 is 50 and another 30 towards positive. So, and then when the membrane potential of this action potential, this is the action potential, this part will reach to +30, then sodium channels will get closed. So all these sodium channels will get closed. So what will happen? No more towards the positivity, but what will happen? The potassium channels will get open. So the potassium will get out from inside the axon to outside. So when the potassium gets out, potassium is also positive. So now positive ions are getting out, right? So what will happen? We'll move back towards the resting membrane potential. Okay. So this is called the repolarization of the action potential. So this is depolarization. Right? And when it will reach to +30, sodium channels will get closed, sodium closed. But +30 will do what? Interestingly, will open the potassium channels. So +30 will close the sodium channels, but will open potassium channels. So potassium will get out, or efflux of potassium will cause the repolarization. So repolarization will take place due to efflux influx of potassium. Makes sense? And we'll get back to this is the resting moment potential. But what happens often, since the potassium channels are getting opened, and when the moment potential returns to the resting moment potential, potassium channels start to get closed but will not be closed completely. Potassium channels take longer time. Potassium channels are slow to get closed, so some more potassium will get out. So that means the moment potential will move further down than the resting moment potential, and this part is called hyperpolarization. Okay, below the resting moment potential. So hyperpolarization. Make sense? So after hyperpolarization, then the sodium-potassium pump and other factors will move the membrane potential back to the resting membrane potential, because if the membrane potential does not return to the resting membrane potential, which is minus 70 here, another action potential will be difficult to generate, right? Because here, for example, if it is minus 90 millivolt, for example, if you present the stimulus here, it will be difficult to move it to the threshold then from here, from minus 70, right? Because this is minus 20 millivolt less. So minus 20 more. So first it has to move to the resting, then to the threshold to produce the action potential. So we'll need very strong stimulus to stimulate the cell. Okay. So now if we see the graded potential again here, this is another neuron, and you see the axon terminal here, okay, and the dendrites. If I give inhibitory stimulus, not excitatory, inhibitory stimulus, then what will happen? This will move the open the potassium and chloride channels. So in this case, inside the neuron, the resting membrane potential is minus 70. Resting membrane potential is minus 70 always, right? Like the other one. But since this is inhibitory stimulus, in this case, the potassium and chloride channels will get open. And what will happen? The potassium will get out and chloride will get in. You know that chloride is negatively charged, right? Anion, and potassium is positively charged cation. So when positive will get out and negative will get in, then inside will become more negative, right? So from minus 70 it can go to minus 80 or minus 90, right? So no action potential, no depolarization will occur, rather hyperpolarization, because it is moving from minus 70 to further negative, like minus 80 millivolt. If more stronger inhibitory stimulus, it can go further down, right, to minus 90 millivolt. But the membrane potential in this case is not going upwards, instead going down towards more negative, right, minus 80 or minus 90. So this is also graded potential, but inhibitory potential. Make sense? So graded potential AC could be depolarization or hyperpolarization. So graded potential could be either depolarization of the membrane or hyperpolarization, depending on the type of stimulus. Okay, but will not reach to threshold, never reaches threshold. Okay, so subthreshold, and will not produce action potential. But action potential, as I mentioned, once the action potential is generated, it will travel far, not localized. So graded potential is localized, but action potential, if I write action potential here, is not localized, can travel far, long distance. Okay, and will maintain the strength, same strength, same, will not get weak. Okay, and only cause depolarization, no hyperpolarization, because depolarization must reach the threshold, right? Depolarization. And action potential usually takes place in the excitatory cells or excitatory membranes. Excitatory membranes. For example, membranes of the neurons or muscles, muscle cells, or muscle fibers. Okay, so graded potential takes place in these excitatory membranes that I showed if the stimulus is weak, as well as in the other cells of the body, non-excitatory cells. Okay, so that's the difference between these two types of potential. Okay, action potential is very helpful to send the signal far, and that's why the receptors produce the action potential to send that signal to the brain. So from the skin to the brain, or from the eye to the brain, from your nose to the brain, the sensory signals must travel long distance, right? So action potential must be generated in those sensory receptors to send the signal by producing action potential. Okay. Now we'll talk about polarization. As you know that in a cell, like this is a neuron, again, so axon terminal, okay, it's the nucleus, dendrite. So now you see that there is a polarization or imbalance is always present in all the cells in your body. In every cell, the polarization is there. What is the polarization? That means if I just take a small chunk of cell membrane from here and see here, inside the cell, in all cells of your body, it is not like outside is 0, inside is, the difference is 0. Always inside is more negative, and that is the polarization. So how that negativity is maintained, which is the resting membrane potential, right? Always negative. In a neuron, it is -70 on average. So how that is created and maintained. So we'll talk about that. That is the resting membrane potential. In resting condition, that polarization is there. Okay. So first, three types of channels are very important, although more than three types of channels work there, but three types of channels are very important, and I will explain those three types of channels. One is here. This is the sodium-potassium pump. Sodium-potassium pump. Pump requires ATP, energy, right? To pump something, you need the energy. So to give that energy, ATPase works here. ATPase, okay, work here on that pump. And what that these pumps do is pushes the sodium out and brings the potassium in. So when the pump is activated, what happens? 3 sodium ions get out. So 3 positive sodium ions get out and 2 potassium gets in. Make sense? Same thing here. 3 sodium gets out. 3 sodium. That means 3 plus is getting out, and next change 2 potassium gets in. Make sense? So now you see here, if inside is 0 before the pump is activated or pumps are activated, 0 millivolt. Since more positive is getting out, less positive is getting in, this will move the membrane potential towards negative, right? So, but these pumps contribute very little to create the resting membrane potential. So, like only a few millivolt from 0 to, for example, -5 millivolt. But we need to bring it down to -70 to create the resting membrane potential, right? So what happens? So this is step one type of channels, and then another type of channels are present in the cell membrane. These are called leaky leaky potassium channels. Always open, leaky. Okay, so what happens inside the cell? The potassium ions, potassium are positive, and these potassium ions are bound or attached to some negatively charged particles like proteins, negative, and phosphate. So phosphates are also weakly negative. So you see that inside the cell, the potassium is bound or attached to proteins, negative, right? Potassium is positive, and phosphate. okay, also negative. And these complexes, the potassium with protein and potassium with phosphate, they start to get out through the leak channel. Okay, so they start to get out. But see, the proteins and phosphate, these are larger molecules. Okay, so when the potassium try to get out together with the proteins and phosphate molecules, the proteins and phosphates, they cannot exit or get out. Potassium alone gets out because the membrane is highly permeable for potassium. So that high permeability of the membrane would let the potassium get out. So now what happens? The potassium, huge amount of potassium will get out because the membrane is highly permeable to potassium. So will get out, but the potassium will leave the proteins and phosphates inside the cell or neuron. So these are negative. So negative ions are now staying, but positive potassium is getting out. So what will happen? The membrane potential inside the cell will move down towards negative because of the remaining proteins and phosphate, and will move down, can move down to all the way minus 90 millivolt. Make sense? Which is less than minus 70. So you see that the membrane potential moves down a lot. Now, so this is number 2, leaky potassium channels. Now, also in the cell membrane, you have the leaky sodium channels. So leaky sodium channels. They also stay open, but less permeable, significantly less permeable. So what happens? So through the leaky channel, when the sodium gets in, you see, sodium is cation positive, so the membrane potential will move a little bit up towards positive. So from minus 90 it may move up to minus 70 millivolt. So this is the actual resting membrane potential, average resting membrane potential in a typical neuron, right? So that's how the resting membrane potential is established, okay, and always negative. So 3 factors are the main factors here. First I'm going over quickly. If you just check cell membrane and see inside and outside, this is outside and this is inside, right? So first, number 1 is that pump, sodium potassium pump, activated by ATPase, that will break the ATP to give energy, and that pump will be activated, which will send 3 sodium out and bring 2 potassium in. So the membrane potential will move from 0 to a little bit towards negative, a few millivolt only. For example, here minus 5 millivolt, okay, down. And then the leaky potassium channel, highly sensitive, significantly more permeable than sodium leaky channels. So what will happen? A lot of potassium will leave the cell. And when the potassium leaves, that means positive is getting out huge amount. So that will lower the membrane potential right inside the cell. Also, when the potassium leaves, they will free the phosphate and proteins. Those are negative molecules. So that will also add further negativity. So the membrane potential will move down a lot. It may move down to, for example, minus 90 millivolt. Now, Number 3 is leaky sodium channels, right? And when the leaky sodium channels would let the sodium get in because sodium concentration is high outside, right? You see, sodium will get in small amount because the membrane permeability is much lower than potassium. So that will, since sodium positive is getting in, from minus 90, the membrane potential will move down to move up actually towards positive to minus 70 millivolt, and that is the resting membrane potential. Okay, so that's how the resting membrane potential is established. Okay. So now, after the resting membrane potential is developed, that is the polarization, difference between the outside and the inside. Inside is always negative a lot. Then if I give a stimulus, I'm going back again to the stimulus. If I give a stimulus which is excitatory, and strong enough to move the membrane potential from minus 70 millivolt to the threshold, which is minus 50 millivolt, then the sensitive channels, voltage gated sodium channels will be activated and will be opened, right? So sodium will get in, and since these are voltage gated, huge amount of a number of channels will get opened suddenly, abruptly. Okay, and inside the axon, this is the axon hillock here, the sodium concentration will go up suddenly, right? And that sodium will flow down, and as the sodium, huge amount of sodium will move down, that will move the membrane potential that will, as the sodium will flow or move down, will open more and more sodium channels along the axon, right? So you know that when abruptly, suddenly, huge amount of sodium channels will get open, the membrane potential will again let me draw here. So this is the resting membrane potential, right, which is minus 70 millivolt. This is the threshold, which is minus 50 millivolt, and when the membrane potential will reach to minus 50 here, that means will reach here, then the voltage gated sodium channels will get open, right? So suddenly huge amount of sodium channels will get open to move it to plus 30 millivolt. Now you see what happens when 30 millivolt will the membrane potential will reach to 30 plus 30 millivolt. What will happen? The potassium channels will get open, that I showed you in previous slides. So here sodium is getting in, and that sodium will move down and will open more sodium channels along the way. Okay, so everywhere plus 30, because sodium is getting in, right? So plus 30. plus 30. Okay, that is depolarization, and that plus 30 will stop the sodium influx. So after depolarization, sodium influx will be stopped, and then plus 30 will open the potassium channels. So potassium efflux or outflux. So the potassium will get out. Makes sense? So when the potassium will get out, we'll get the repolarization. So the membrane potential will go back to resting, and as I told you that the potassium channels take some little more time to get closed, so it will move down further to produce hyperpolarization before it goes back to resting again. Okay, now when the depolarization takes place during this time, action potential is generated, right? This is depolarization and this is action potential here, and that would open calcium channels here near the axon terminal or axon terminal buttons. So calcium channels will get open. Now you see here what will happen. The calcium will get in. Okay, so sodium, huge amount of sodium influx will also open the calcium channels near the axon terminal. Okay, and this calcium influx is very important. Why? Because in the axon terminal or presynaptic terminal, you have the vesicles filled with the neurotransmitters. Okay, so the calcium will get in and will move the vesicles to the presynaptic membrane, and you have special proteins here. So the vesicles will move and will bind with the protein, and that will cause the rupture of the vesicles. So the vesicles will rupture, will first get attached to the proteins here and to the cell membrane and axon terminal, and will get ruptured. Okay, so that is the exocytosis. Exocytosis, that's the rupture of the vesicles to release the neurotransmitters. Make sense? So what kind of neurotransmitters will be released? That depends on the neurotransmitters stored in these vesicles, and what neurotransmitters are stored, that depends on the number of the type of neurons. Okay, so what happens next? Immediately after the calcium influx takes place due to influx of sodium, a huge amount of sodium inside the axon terminal, then as you know that sodium channels will get closed when the membrane potential reaches plus 30. Then that will also close the calcium channels. So calcium channels will get closed. Calcium channels will get closed. So no more calcium will get in, no more rupture of the vesicles. Makes sense? So the rupture of the vesicles will take place at once, and release of neurotransmitter at once, but immediately after it will be stopped. It will not release anymore because the calcium channels will get closed when the membrane potential reaches to plus 30. Okay, so plus 30 is doing what? Plus 30 is opening the potassium channels to cause the repolarization, opening of potassium channels and closing of calcium channels. Make sense? So opening of potassium will cause repolarization, and closing of calcium will stop exocytosis or stop release of neurotransmitter. Okay, here, and minus 50, which is the threshold millivolt, is doing what? We activating the voltage gated sodium channels. So opening voltage gated sodium channels. So sodium influx suddenly, and that will cause the action potential. Okay. Okay, now let's see a term that is called summation that takes place in the synapses. So this is a neuron, and this is a synapse, right? So this is the synapse. And if this neuron is excitatory neuron, so this is excitatory, right? So when you stimulate this neuron from the axon terminal, you see here neurotransmitter, you already know how the neurotransmitter is released. Since this is an excitatory neuron, the neurotransmitter that will be released is excitatory neurotransmitter. The most powerful and common excitatory neurotransmitter is glutamate. So let me write glutamate here. Glutamate, okay, will be released, and this is the postsynaptic membrane of the next neuron, right? So the glutamate will excite the postsynaptic neuron. So this neuron will be excited, right, by that glutamate excitatory neurotransmitter. Okay, and if we see here how the summation takes place, I have to draw the same thing here. So this is the resting membrane potential, okay, minus 70 millivolt. This is the threshold here, for example, minus 50 millivolt, right? So threshold. So in this neuron, when it will be excited, the membrane potential will move towards positivity, right? That means depolarization will take place. However, if the stimulation here or stimulus is not very strong, as I mentioned before, it may not reach to the threshold, right? It will be stopped here. But if you see, I repeatedly present the stimulus, even the same stimulus that could not move the membrane potential to the threshold, subthreshold membrane potential. If I present it in a short period of time, repetitive stimulation or burst of stimulus, that means many stimuli, weak stimuli together, one after another, not together, one after another, repeatedly, like stimulus is here. This is a single stimulus, will move up to a certain limit. But if I repeatedly present the stimulus, this is called the burst of stimulus, then what will happen? We'll see summation. The first stimulus will move up to here, the second one will move up to here, the third one will move up further, and it will reach to the threshold, right? So once it reaches the threshold, then you don't have to think, it will produce action potential by itself, right? So this is called the temporal summation. Okay, summation could be two types: temporal summation and spatial summation. So temporal temporal summation. Makes sense? I'm presenting repetitive stimuli one after another, so they will be added to move the membrane potential to the threshold. So here only one excitatory neuron, and this is the neuron receiving that signal. This is also one neuron, so one on one, right? Single stimulus won't produce action potential, but if I present multiple stimuli in short period of time, that is temporal summation will produce action potential. Now, another way summation can occur. This is one excitatory neuron. This is the postsynaptic neuron. So here, this is presynaptic neuron. So if I present stimulus here, it will not produce the action potential, like it will move up to here. But if convergence occurs on this neuron, for example, this neuron is getting signal from all these three neurons, although individually, like this one, if it produces excitatory, this is excitatory 1, this is excitatory 2, this is excitatory 3. If I individually activate these neurons, excitatory neurons, they will always produce some threshold response. Makes sense? But if I simultaneously together present a stimulus in these three neurons, then what will happen? The convergence or summation will take place, and that will move it to the threshold and will cause the action potential. And this is called spatial summation, multiple to one. Okay. Okay, now I'll talk about the sensory receptors. Sensory receptors could be two types. We can classify them two ways actually: chemical, two ways, receptors anatomical, that means structural, and functional. Okay, anatomical is based on the structure. We know that the receptors are housed inside the structures or organs in the body, sensory organs. So if you see some receptors, this is a nerve. For example, this is your skin. You will see some receptors are present at the free nerve endings. So these are the receptors attached to the free nerve endings. So they are easy to activate, right? To activate them, weak stimulus will be able to activate them. For example, pain receptors are present at the free nerve ending. So these are free. Some receptors are housed inside the capsules or membranes. For example, in your skin, you have touch receptors, right? And touch receptors could be two types. One is the soft or fine touch, another is the deep touch or crude touch. Okay, so for example, Pacinian corpuscles, and you'll see the receptors are housed or located inside the membranes or bounded by the membrane. So these are called corpuscles. Okay, so this is located in the superficial part of the skin, and also we see the membrane bound receptors in the deeper part of the skin. So this is, for example, dermis, this is epidermis. So inside this corpuscle you have the neurons, but even if you see the corpuscles, in this case, if I see inside, I'll see that the membrane bound receptors are located inside the sac-like structures like this. Many sacs or fluid-filled sacs are stuck together, and when you touch the skin, that will cause the sliding movement of these sacs. So these sacs will move to opposite directions, and that friction will cause the activation of the receptors. Makes sense? So these are the receptors, and the movement of the sacs, fluid-filled sacs, will activate the neurons. So this is one type of corpuscle, and this is another deep part of the skin. You'll see this is called Pacinian Corpuscle. So in this case, we see that free nerve endings are inside, but these are not actually open. Here, if I see, I'll see that many layers, like onions, are protecting these free nerves endings. Okay, so the receptors are attached to the free nerves. So in this case, they are free, not membrane bound. But here, you see the many layers of the membrane is protecting the receptors. So some receptors are more free, some receptors are heavily protected, and they are activated by different types of stimulus, right? If you see the receptors in other organs, for example, the retina of the eye, the receptors are the photoreceptors, right? And they are heavily protected by the facial and cranial bones, right, inside the orbital fossa, as well as inside the eyeball. So heavily protected. Also, the hair cells, the cochlear hair cells are heavily protected, right? So the photoreceptors, sound receptors, or hair receptors are heavily protected. Cochlea is in the inner ear, protected by the temporal bone. So heavily protected. But some receptors are not that heavily protected, located on the body surface. For example, your skin receptors, as well as olfactory, right, inside the nose, not heavily protected. Always we get a chance to get infection inside the nose, or mechanical friction. So olfactory, not protected. Also gustatory or taste receptors are not protected on the surface of the tongue, and when you eat something, some receptors could be destroyed, or when you eat hot food or drink, that can also kill some receptors. So you see, these are not well protected. Okay. So anatomically, the receptors could be located in different ways. Functional classification, you know that different receptors are stimulated by different means. Some are activated by mechanical pressure, like touch or pressure or vibration receptors, right? Or even the hair receptors of your cochlea, sound receptors. Those are activated by pressure. Mechanical. Some are activated chemically, like the taste receptors or olfactory receptors, smell receptors, chemical, right? Some receptors are activated by the electromagnetic energy, like light. Okay, photoreceptors. Some receptors are activated by the movement, like proprioceptors. Right? And proprioceptors are present in the muscle spindles and the joints, so they help you to locate the body parts, positions of the body parts in 3D space. So when you move the body part, the receptors are activated to tell you the location of your hand or foot or other body parts. Okay, so some receptors are activated by motion, like acceleration, movement of the head. You know that the receptors in the semicircular canals, vestibule, so vestibular receptors or semicircular canal receptors by the movement of the fluid inside the vestibule and semicircular canal. So you see that functionally different types of receptors are present in the body. So I have posted a chart where you can see the types of receptors and the locations and how they are activated. Okay, so now the last thing is I'll talk about the cerebral cortex. In the cerebral cortex, you have many functional areas, right? First, I'll talk about this, then I'll talk about temporal and spatial coding, functional areas. So I'll just go over the important functional areas, then when we'll talk more about individual sensory systems, we'll talk more detail. So this is the brain. Okay, and in the brain surface you'll see many grooves. Some are shallow, some are deep grooves. Shallow grooves are many, and the deep grooves are only few. So I'll just show you the deep grooves here. This is one deep groove. This is called the central sulcus. Central sulcus. So the grooves are sulci. One singular is sulcus, sulci is plural. So central sulcus is very important. Why? Because it is separating the frontal lobe and the parietal lobe. And then you have another groove here. This is called the parieto-occipital sulcus. Parieto-occipital, okay, sulcus. And this one, you see, this is called the lateral sulcus. Lateral sulcus. Okay, so these are three important, and this is the temporal lobe, this is the occipital lobe, this is the parietal lobe, and this is the frontal lobe. Now, in front of the central sulcus, you have a very important functional area here. This is called the primary motor cortex. Okay, and in front of the primary motor cortex, you have the motor association cortex. And motor association cortex has two areas, functional areas. One is called the supplementary motor cortex. So let me say here supplementary motor cortex and pre-motor cortex. So primary motor cortex, and in front of the primary motor cortex you have the premotor cortex and supplementary motor cortex, and these two together is called the motor association cortex or area. Okay, so primary and association. Primary motor cortex controls the voluntary movement of the body. So from the left side of the brain, from the primary motor cortex, the signal goes to the right side, and from the right side of the brain goes to the left side of the body to control the movement of the opposite side of the body. Okay, so that is the function of the primary motor cortex, voluntary movement, okay, of the opposite half of the body. And the association cortex, which includes the premotor cortex and supplementary motor cortex, these two cortices helps you to plan the movement. So after the planning, then the signal will go to the primary motor cortex, and primary motor cortex will execute the function. Okay, so analyzing and planning is the function of the association cortex. Then if you move further towards the front, this is the frontal eye field. Okay, so frontal eye field is important because this area controls the movement of the eyeballs. So your eyeballs movement are controlled by the frontal eye field. Okay, and then this whole area is called the prefrontal cortex. Very important. Higher brain functions, for example, your ability to judge, your reasoning, your memory, your problem solving, all those higher order brain functions are actually controlled by this area. So interpretation or analyzing, interpretation of something, problem solving, for example, math problem solving, your judgment, your emotion and memory, okay, reasoning, all those functions are higher brain functions controlled by the prefrontal cortex. Okay, and here you have another area that is called the Broca's area. Which is a motor area, and this motor area is responsible for the speech, production of speech. So it sends signal to the vocal cords. Okay, now this is the temporal lobe. So if you see the upper part of the temporal lobe near the bank of the lateral sulcus, this is the primary auditory cortex. So the frequency, pitch of sound, the intensity of sound, okay, sound perception is given by the primary auditory cortex. Okay, and then around the primary you always have the association. So this is the association auditory cortex. Make sense? Auditory association cortex. And this one gives you the perception by analyzing that sound received by the primary auditory cortex, and would tell you if it is a human voice or a call of a cat or a dog or something else. So what sound is that? That is actually helped by recognition is helped by the auditory association cortex. So association cortices are mainly helping in recognizing, and primary cortices are mainly giving you the awakening signal. Okay, and then if you see the medial side, this is the lateral side. If you go more towards inwards, deeper, you have the olfactory, primary olfactory cortex, that means smell, more towards the medial side, and you have olfactory association cortex too. Here, olfactory, always primary, around the primary you have the association. So primary olfactory cortex would give you the smell, perception of smell or awakening signal of smell. Then the association cortex would give you more information about that smell. Okay, if you smelled that before, or is it a good smell or a bad smell, based on your memory, all those things are processed in the association, olfactory association cortex. Okay, so those are the important functional areas in the temporal lobe. Now let's talk about the parietal lobe. This is the central sulcus again, and posterior to the central sulcus, this is the primary somatosensory cortex. So the touch signal, your tactile signal, would be processed here. Okay, will be received, and you will get the signal that something touched your body, and which area of the body got the touch. Then you have the auditory, somatosensory association cortex. Somatosensory association, again, association is next to the primary, right? Somatosensory association cortex will help you to recognize what kind of touch is that. Okay, so recognition by the association cortex, and awakening signal will be processed in the primary cortex. Now behind or posterior to the somatosensory association cortex, this area behind it is called this is a multimodal area. Posterior, this multimodal area is posterior parietal cortex. Okay, and posterior parietal cortex is a multimodal area. Why it is called multimodal? Because in this area, your auditory from here, visual from here, and somatosensory from here, all are combined. So multimodal area, and gives you the perception. When you learn something in anatomy, like if I give you the heart model, you will touch it, so somatosensory signal will go from your touch area, right, tactile area, and you see at the same time the shape and size, the color, the signal from the visual will go there. And if I tell you this is a heart, this is the shape, that sound will go there, and all those should be combined to give you help you to learn, right? And we all know that we use all those modalities together to learn the things, right? So that's the posterior parietal cortex, multimodal area. And now this is the occipital lobe, and all the way back to the occipital lobe, this is the primary visual cortex. And around the primary visual, you have the visual association cortices. There are many, and they receive signal from the primary. Primary first receives the signal from your retina and then give to different association areas. For example, one area will process the color, another area will process the motion, another area will process the texture or shape of those things, right? So that's why you have many association areas around the primary visual cortex. Okay, so this will give you some idea how the cerebral cortex has different functional areas. Now, if I just cut and take a chunk of piece of cortex out from here, I'll see how many layers under the microscope. There are 6 layers. Okay, so 1, 2, 3, 4, 5, 6. This is the surface, this is deep, and area 4, that is layer 4. This layer is very important because this layer is called the input layer. So signal, from thalamus or other subcortical areas will be received by these neurons. So the signal will be first received by layer 4. Okay, this layer. So this is the input layer. And then from here, the signal will go to layer 5 and layer 6, as well as signal goes horizontally to the other neurons within the layer. So downward signal will go to layer 5 and layer 6 neurons, and these neurons are output neurons. That means will send the signal downwards, descending. So ascending signal will be received by the neurons in layer 4, and from layer 4 the signal will go to the other layers, as well as the output layers 5 and 6 will receive the signal, and the signal will get out. Okay, so 6 layers in the cortex, and the cortical layers are heavily filled with the neurons, the cell bodies of the neurons, right? So that's why it is called gray matter. It will look gray, not white. White is in the deep part under the cortex. This is the white matter formed by the fibers, myelin sheet of the fibers. Okay, so that's about the cerebral cortex, and the last thing is the coding. Temporal and spatial coding. Those are two important types of sensory codings that take place in the brain. I'll just give you a couple of examples of each type of coding. Okay, so spatial and temporal coding. First, temporal coding. You know that the sensory inputs enter into the brain from different events that are happening outside of the body, right? So if you are listening an orchestra, then many people play different instruments simultaneously, right? And they play together, and that gives you a very good perception of that event. However, if all players, those who are playing the instruments, they randomly play the instruments, then that will make no sense. It will be like a noise, right? So the things that enter into your brain should maintain the time. The appropriate timing is very important, right? So from all the instruments, the signal should go following the appropriate timing. That's the temporal coding. And spatial coding is the location that receives the signal from the event outside of the body. For example, if 2 points touch your skin, like if you use a slide caliper and there are 2 pointed ends, and if you touch your skin surface by 2 without looking, you can feel that, right? But if you bring them very close to each other, then you won't be able to discriminate, right? So the signal that goes to the brain from the receptors here can tell the brain that you have 2 points or 1 points, right? So up to a certain limit, the brain can discriminate. That is called 2-point discrimination. Okay, so that is a good example of spatial coding. Another is like you have 2 eyes, right? And when you look at something, your 2 eyes receive the image slightly differently. That is called the binocular disparity, right? Because your eyes are away from each other, right? Slightly apart. So the picture that falls on your right and left eyes are slightly different, and your brain receives that signal, and by interpreting that signal, your brain can tell the distance of that object. So that is another very good example of the spatial coding. Another is like this is your head, and these are your 2 ears, and you are listening a sound that is here, the source of sound. Now, in this case, the distance of your right and left ears is same, right? So no difference. So they will arrive, the sound will arrive in your right and left ears at the same time. So the difference is 0. Now, if I move it to here, the sound source is now a slightly different location in space. Now, from this source, the sound will arrive in your right ear faster than or earlier than your left. So your brain can interpret that difference, and by interpreting that difference, your brain can tell the location of the sound source in space. So that's the spatial coding, right? In different locations in space. So that's it. Let's stop here.